My name is Nur Akira Natasha binti Abdul Ghaffar, student ID 2020605.26. So today our group will explain about chapter 6, ovulation. So what is ovulation? Ovulation is actually a physiologic process defined by the rupture of Graafian follicle and release of secondary oocyte from the ovary into the fallopian tube for fertilization to occur. If the egg is not fertilized within a certain interval of time, the egg will disintegrate and be shed along with the menstrual blood. However, if fertilization does occur, the uterus will prepare for implantation and pregnancy. In mammalian, there are two modes of ovulation, which are induced and spontaneous ovulation. In induced ovulation, the ovulation is stimulated by a physical or a sexual behavior by a male partner. Meanwhile, in spontaneous ovulation, the ovulation occurs cyclically regardless of any stimulation by a male partner. Example of induced ovulators are cats, rabbits, and alpaca. Meanwhile, example of spontaneous ovulators are humans, cows, and mice. The key differences in distinguishing between induced and spontaneous ovulation. In spontaneous ovulators usually have a constant or a regular cycle of hormones and ovulation regardless of any stimulation by a male partner. Meanwhile, induced ovulators, they usually do not have any constant cycle for ovulation.
Early in the follicular phase, GnRH and active and stimulate the release of FSH from the anterior pituitary and augment FSH action in granulosa cells by increasing FSH receptor expression and increasing aromatase activity. It begins on the first day of period and ends when ovulation occurs. The average length of the follicular phase is 16 days, but it can last anywhere from 11 to 27 days depending on cycle. The ovary is least hormonally active with the lowest levels of estradiol and progesterone production during this time of the menstrual cycle. FSH continues to stimulate folliculogenesis and aromatization of androgens to estradiol in the ovarian follicles. Increased estradiol inhibits GnRH release from the hypothalamus by negative feedback, which leads to a decrease in FSH. The decrease in FSH is also a result of rising inhibin levels. A dominant follicle is selected at approximately day 7 of the cycle. Estradiol release continues to increase from the granulosa cells of the dominant follicle, and this stimulates the proliferation of uterine endometrium, increasing the number of glands. A sustained rise of estradiol production from the dominant follicle leads to a temporary switch from negative to positive feedback of estradiol on the hypothalamus and pituitary, with rising estradiol levels now increasing LH production rather than inhibiting it. Late in the follicle phase, FSH induces LH receptors in the granulosa cells, which stimulates progesterone production. This rise in progesterone production and stagnation augments the positive feedback effect of estradiol. The positive feedback of estrogen and progesterone results in the mid-cycle LH surge, the critical event for ovulation.
There are three mechanisms that help in ovulation, which are endocrine, neurobiological, and neuromuscular mechanisms. When the positive feedback mechanism happens, it will result the LH surge that will play an important role in cumulus expansion, protein synthesis in and around follicle, and increase the blood flow to ovary and follicle. Firstly, cumulus cell will produce hyaluronic acid and protein, resulting in cumulus expansion. This will make the cumulus and oocyte separate from granulosa cells. Next, the blood flow will increase as the result from the increasing of PGE2 and histamine local response. So, the vascular permeability will be increased and produce edema in tissue surrounding follicle and increase plasminogen. This will maintain the positive follicular pressure and will drive the force for extrusion of oocyte. Besides, LH surge can cause protein synthesis and cellular differentiation around follicle. Steroid biosynthesis shifts to production of progesterone, which will trigger the production of plasminogen activator from granulosa. Progesterone will also trigger production of inactive collagenase from theca interna. Plasminogen activator will help in the conversion of inactive collagenase to active form that will help in weaken the collagen of tunica albuginea and theca layer. Then, granulosa cell will increase the production of follicular fluid and stigma begins to push out and weakens, resulting in the follicular rupture.
Maturation of oocyte or development of oocyte is also known as oogenesis, in which an oocyte transforms into a mature ovum. It consists of different phases such as primordial germ cell, oogonia, primary oocyte, secondary oocyte, oocyte, and then ovum. Primordial germ cells are embryonic cells that have the potential to develop into sperm or oocyte cells. These cells migrate toward the testicle or the ovaries. Once there, they will be influenced by the cell's death to develop into oogonia. The organism multiplies during the first 5 months of prenatal development through a process called mitotic cell division. Only young sperm and egg cells exhibit it. The egg will go through two distinct mitotic cell divisions, resulting in the growth and maturation of the oocyte. The oocytes stop increasing in number and start to mature individually at the end of prenatal development. This is their first mitotic cell division. This cell division results in oocyte growth. They do not, however, immediately advance from childhood to adulthood. The primary oocytes stop developing and stay that way until the next stage is triggered by reproductive hormones. At the age of puberty, oogenesis will continue. The following phase of oocyte maturity is accelerated by puberty. Over the course of a woman's reproductive years, they alternately develop. A fresh batch of primary oocytes starts to mature each month. A primary oocyte completes stage 1 of mitotic cells divisions once it is influenced by reproductive hormones. Eventually, the little polar body degrades. The subsequent stage of maturation starts with the secondary oocyte. Second stage of mitotic cell division starts within the oocyte. The secondary oocyte will eventually divide once more into two distinct cells, a smaller polar body cell and a bigger mature cell. An oocyte is a term for this more developed bigger cell. The smaller polar body cell will eventually degenerate as before. When the oocyte reaches the oocyte stage of development, ovulation takes place. And this is the structure of oocyte, which generally consists of corona radiata, zona pellucida, vitally membrane, germinal vesicle, and germinal spot and ooplasm. In conclusion, we have learned about ovulation in an interesting way by doing some research and making this video. We have learned how complex and beautiful the process of ovulation is. We hope that whoever watches this video will gain new knowledge and share it with your friends. Thank you for watching.